biological classification. After studying the living world, we will now study the second part of this subject, biological classification. So we have studied about genus, species, taxonomic hierarchy, and what are the aids for taxonomical aids. In biological classification, we study about the taxonomy itself, the different systems of classification proposed. But out of all many of the systems which are proposed by the scientists, we now follow only one type of classification. Now, just a brief aspect about the history of classification. It was Aristotle who first classified living organisms into plants and animals. So plants and animals were first classified separately by Aristotle. And after Aristotle, other scientists, they also classified. Now when Aristotle classified plants and animals, now plants means organisms which are having stem, root, leaf, and they are having the property of photosynthesis and all that. But animals, when we say animals, we think that they are having the power of locomotion. So based on that he classified. And further he also classified plants into three groups, like herbs, shrubs and trees. And animals also he classified as aquatic, terrestrial and arboreal. Now after classification of proposed by Aristotle, there was one confusion or a problem. There were some organisms which had neither plant character nor animal character. That means when we observe that organism, we cannot perfectly say that it is a plant or animal. So that was one drawback in Aristotle's classification. So that is why after Aristotle, when he divided that living organisms into two kingdoms, he called it as kingdom plant and kingdom animal. But after that, Ernst Haeckel, a German scientist, he classified organisms into three kingdom classification: protista, plants, animals. So that means organisms which are having both plant and animal character, or which were neither having plant or animal character, he classified them into protista group. And then that was also another problem when Ernst Haeckel he classified this. So that is why another scientist called as Copeland, he classified organisms into four kingdoms. So that is monera, protista, plant, animal. So he divided monera as a separate group and he classified organisms into four kingdoms. Living organisms were classified into four kingdoms. Then after Copeland, there was one scientist called as Robert Whittaker. So Robert Whittaker, in the year 1969, he proposed the 5 kingdom classification. He divided living organisms into 5 kingdoms. So those 5 kingdoms, what he classified were monera, protista, fungi, plants, which he called it as plantae, and animals, that is Animalia. So these are the 5 kingdoms what he classified. So we are following Whittaker's system of classification. We are not bothered about Aristotle's classification, Ernst Haeckel's 3-kingdom classification, Copeland's 4-kingdom classification, all that. But that is about the history of classification. We are following according to our syllabus, or in general, universally this is followed in classification. But however, after Whittaker's classification which he proposed, there is also 6-kingdom classification which is proposed, and even recently 7-kingdom classification is also proposed. So that means they have divided monera into 2 groups again. That is why the number of kingdoms will increase. But however, we are not concerned, but just for an information, other than, means this is not the only classification which is there existing. And after 5-kingdom classification, there is also 6-kingdom classification, there is also 7-kingdom classification. Now we shall be more concerned, or we are more interested in this Whittaker's 5-kingdom classification. So Robert Whittaker, actually an American ecologist, he is more interested in ecology. At the same time, he also studied about classification, and in ecology also he has contributed significant ideas in ecology. So he is an American ecologist actually. He proposed this 5-kingdom classification which we are more concerned with. So he divided organisms into 5 kingdoms: monera, protista, fungi, plantae and animalia. Now one by one in this chapter, biological classification, we are studying 3 kingdoms, that is their characters and some examples of them. Monera, what are the characters of monera, protista, then fungi. So these are the 3 things what we are going to study. Now this coming to one by one the monera, protista and fungi. So plant we will be studying in the next chapter. Animal, it is also another chapter. So this is the 3rd chapter, this is the 4th chapter. So this is about the history of classification. So now Whittaker's classification: monera, protista, fungi, plantae and animals. Now monera, this is a group which includes what are called as prokaryotes. So there is one more just a general classification where organisms are classified into prokaryotes and eukaryotes. So all prokaryotes are included under monera. So monera is the group which includes all prokaryotes. And one more thing is, this prokaryotes are unicellular organisms, always unicellular. We never find multicellular organisms in monera. All examples, organisms which are included under monera, they are prokaryotes, unicellular prokaryotes. The remaining 4 kingdoms, they are eukaryotes. So this is eukaryote. All these are eukaryotes. Protista is also eukaryote, fungi, plants and animals. So we are eukaryotes. Now what are prokaryotes and eukaryotes? Next part. So prokaryote and eukaryote, just to explain the difference between the cell of a prokaryote and a eukaryote. Suppose this is a prokaryote and this is an eukaryote. What are the differences between the cell of a prokaryote and a cell of a eukaryote? So this is just some basic idea or a thing which we have to understand. Now usually the cell shape, what I have drawn a square, so there are different shapes of cells. Then here in prokaryote the nucleus is not definite. So that means indefinite nucleus. Now whatever the nuclear material, the DNA or the nuclear material, it is freely present in the center of the cell or in the cytoplasm. So this nuclear material, which is also sometimes called as nucleoid or indefinite nucleus or incipient nucleus sometimes. So this is found in the prokaryote. But in case of eukaryote, this nuclear material is surrounded by a well-defined nuclear membrane. So but in case of prokaryote, you don't come across such kind of nuclear membrane. It is absent purely. This is one main difference. So such nucleus is called as definite nucleus, a perfect nucleus. So this is indefinite nucleus or sometimes referred as nucleoid. And another difference between this nucleus, indefinite and definite nucleus is, in case of nucleoid, the DNA which is present, it is usually pure DNA. That means it is not associated with any type of material. But in case of definite nucleus, as we are all, so that means the remaining four kingdoms are all eukaryotes. So this nucleus which is present, that contains the DNA, it is associated with proteins like histones and non-histone proteins. So or they are also called as nucleoproteins. So such kind of nucleus is present in case of this eukaryotes. So here also there is the DNA, here also there is DNA. This is not surrounded by nuclear membrane. That means it is a naked nucleus. It is also called as incipient nucleus. So it is freely present in the cytoplasm. So this is all the cytoplasm. So this DNA is freely suspended in the cytoplasm. But here it is well protected or surrounded by the nuclear, well-defined nuclear membrane. And also the DNA is associated with the nucleoproteins. But here the DNA is not associated with proteins. But at the most, there may be few proteins. So it is considered as almost absent. So that is why this is indefinite nucleus, this is definite nucleus. This is the main difference between prokaryote and eukaryote. So any prokaryote cell you take, it will be having indefinite nucleus, incipient. Any eukaryote you observe, we will be having definite nucleus. So whatever the shape of the cell or like that it may be. And in addition to this, there are also other differences between prokaryote and eukaryote. So this prokaryotes, they are having incipient nucleus. This is the main character of this. Here all these are having definite nucleus. And other characters when we come across. In this prokaryote, mitochondria, Golgi complex, endoplasmic reticulum, they are absent. So here mitochondria and Golgi complex and endoplasmic reticulum, so these are absent in this, but here we find this in the eukaryotes. This is also another important difference between prokaryote and eukaryote. So for mitochondria, they are the important organelles of the cell. Golgi also, they are important organelles. Endoplasmic reticulum, they are completely absent. They are not found here in the this prokaryote. Then other character is ribosomes are there in both. But the type of ribosomes here, they are of 70 S ribosomes. These are about 80 S ribosomes. So 70 S ribosomes, 80 S ribosomes. So these 70 S and 80 S, more about that we will be studying in the cell chapter when we study the organelles of the cell. Now what is this 70 S and 80 S? Now just in brief to give an idea here. So ribosomes, they are having a property of sedimentation coefficient. So that sedimentation coefficient the property of sedimentation, it is measured in the units of Swedberg units, which is written as S. So when ribosomes are subjected to ultracentrifugation, their sedimentation coefficient is measured in terms of Swedberg units. And so ribosomes which are having 70 Swedberg units, 70 S ribosomes, they are present in the prokaryotes. And ribosomes which are having 80 Swedberg units, they are present in the eukaryotes. So like that there are different types of ribosomes, but however the functions are same. So these are only the property of sedimentation, they differ, but function is as same. Usually the function of ribosome is protein synthesis. They synthesize, they help in the protein synthesis. So that is found in both type of cells. And when we come to another character, cell wall, here in prokaryotes, the cell wall which is present, it is not made up of cellulose. So whenever we speak basically about a cell, we believe that usually the cell wall contains cellulose, but cellulose is absent in case of prokaryotes. And here instead of cellulose, there is another material which is called as peptidoglycan or sometimes mucopolypeptides. So they are but not a cellulose material. So that is cell wall composition is different in prokaryotes and eukaryotes. In case of eukaryotes, if present, it is usually cellulose or pectin or like that, or lignin like that in case of plants. But sometimes when it may be absent, in case of animals you don't find cell wall, and hence there is no question of the cell wall material. But when it is present, it is usually cellulose in case of eukaryotes. So these are the basic differences or the main differences between prokaryotes and eukaryotes. So all examples which we come under monera, they are prokaryotes. So when we speak about prokaryotes, it is definitely coming under kingdom monera. But when we speak of eukaryotes, it may be protista, it may be fungi, it may be plant, or it may be animal. So like that eukaryotes may be included or are included under all these four kingdoms. Now coming to monera again. Now we have understood what are prokaryotes and eukaryotes. So basically we should know, that is usually asked for 1 or 2 marks question, what are the differences between prokaryotes and eukaryotes. So already I said main is the nucleus, then mitochondria, Golgi, endoplasmic reticulum absent, and here it is present. Ribosomes are of 70S and ribosomes are of 80S. Cell wall, it is not made up of cellulose, it is made up of this peptidoglycan, and here it is made up of, if present, it is made up of cellulose usually. And monera, what are the examples coming under monera? So in monera we come across bacteria, and we come across archaebacteria, and we come across cyanobacteria and mycoplasma. So these are the examples of kingdom monera. Now this one by one briefly about bacteria, archaebacteria, cyanobacteria and mycoplasma. These are the examples which we come under. Now when we say bacteria, Again, there are different examples of bacteria. Bacteria means it is a common term used for a group of organisms. Similarly, archaebacteria, it is also a common term. Then similarly, cyanobacteria, mycoplasma. Now bacteria, when we say, it includes different examples of bacteria. For example, lactobacillus, you might have heard, the one which is present in the curds and which helps in the conversion of the milk into curds. And you might have heard about rhizobium bacteria present in the root nodules. That is also a name of a bacteria. So like that, there are different bacteria. Bacteria word is a common term. Just like, for example, insects, when we say, insect means it is not a particular example. It is a common term used for a group of organisms. So an insect may be an housefly, it may be a mosquito, it may be honeybee, it may be silk moth. So like that, there are different types of insects. So coming to the bacteria and studying in common about bacteria. Now bacteria, based on their shape, there are 4 types of bacteria. So if you consider the shape of the bacteria, so there are different shapes in bacteria. Again, suppose if the bacteria are round, they are called as coccus bacteria. If the bacteria are rod shaped, they are called as bacillus bacteria. If the bacteria are spiral, they are called as spirally bacteria. If the bacteria are somewhat comma shaped, they are called as vibrio bacteria. So bacteria which are round like this, bacteria which are rod shaped like this, bacteria which are spiral shaped like this, or sometimes coil shaped, and bacteria which are comma shaped or somewhat half moon shaped, they are called as vibrio bacteria. So this are the 4 types of bacteria. So sometimes this is asked for 2 marks. Classify bacteria based on their shape. Coccus, round bacteria, means spherical. Then rod shaped, bacillus, spirally, they are coil shaped, spiral, then comma shaped or somewhat half moon shaped, they are called as vibrio bacteria. So this is based on the shape of the bacteria. So in common, so the lactobacillus is rod shaped, so that is which is present in the curds, and these other bacteria, they are found in a different type of environment. Now again about bacteria, bacteria are referred as cosmopolitan. So cosmopolitan means universally occurring. So bacteria are found in soil, bacteria are found in water, bacteria are found in air, and sometimes bacteria are also found in some extreme climatic conditions, like this cold ice, that means in the polar regions and even in the higher temperatures. So like that, bacteria are found in all types of climatic conditions, that means soil, water and air. This is about bacteria classification. Now there is one more classification of bacteria based on nutrition. This is one more types of classification. Again there are two types of bacteria: autotrophic bacteria and heterotrophic bacteria. So here there are four types based on the shape, and here they on the based on the nutrition they are mainly two types: autotrophic and heterotrophic. So autotrophic means auto, self. They are capable of synthesizing their own food material using inorganic substances. So using inorganic substances, they are capable of synthesizing their food material. So they prepare their own food material, or they obtain their nutrition from these inorganic substances. Now this inorganic substances are different for different bacteria again. Now in autotrophic again we come across two types. Suppose if the bacteria is having chlorophyll, and this bacteria which is having chlorophyll, it is capable of conducting photosynthesis, and that is said to be photosynthetic bacteria. So that means using carbon dioxide, water and light energy, it will conduct photosynthesis and it will prepare its own food material. In case of another type of bacteria which are also autotrophic, they are called as chemosynthetic bacteria. So in case of chemosynthetic bacteria, so the word itself, so here photo means light. With the help of light it will prepare its food material, and here chemosynthetic, chemicals. So they convert chemical into from one form into another form, or in other words they oxidize chemicals. So they involve in the oxidation process from converting one form of chemical compound into another form, and thus it is called as chemosynthetic bacteria. So that means they obtain their nutrition from these inorganic substances. So when they use these inorganic substances, so they obtain that, they are called as autotrophic bacteria. But there are bacteria which cannot synthesize their food material. So they depend upon nutrition, they depend upon food from on external source. It may be animal or it may be some dead or decaying material, external source. And so that means they do not synthesize their food material. So they are capable of synthesizing from inorganic, and they obtain the food material or they get their nutrition. But heterotrophic are not capable of synthesizing their food material, and they depend for nutrition on the external source. So again here we can classify into two types. Suppose if they depend on the decaying matter, they are called as saprophytic bacteria, and if they depend on other living organisms, they are called as parasitic bacteria. So saprophytic bacteria and parasitic, so that is one type of classification. Now again here under heterotrophic bacteria, so these bacteria when they depend also on the dead and decaying material, they are called as saprophytic. So they depend on other living organisms, they live on other living organisms, they are called as parasitic bacteria. Then these bacteria are again classified into two types. Sometimes bacteria are beneficial, they are useful, and some bacteria they are harmful. So there are beneficial bacteria and there are harmful bacteria. We also get some benefit from bacteria. So just now we have mentioned that milk is converted into curds. It is benefit, isn't it? Now the rhizobium bacteria, which is present in the root nodules of leguminous plants, that is also benefit. It helps in the nitrogen fixation. See here we got two benefits, which in our home we see that milk is converted into curds. That is a common example of bacteria, useful bacteria, uses of bacteria, benefits of bacteria. Then so this milk is converted into curds, nitrogen fixation. Then there are certain bacteria which are producing antibiotics. So production of antibiotics is also another use of bacteria. There are certain species like streptococcus bacteria. It gives an antibiotic called as the streptomycin. Antibiotics are chemicals which are obtained from the living organisms and used to control the population of another living organism. So usually when we are not feeling well, when we visit a doctor, they give certain antibiotics like ampicillin, chloromycetin, or any other chloramphenicol, or many other antibiotics are there. So this is to control the growth of the microorganisms. So the bacteria or any pathogenic organism which is present in our body, their growth is controlled by taking the antibiotics. So antibiotics are chemicals which are obtained from one living organism and used to control the growth of another living organism. That is one another benefit of this bacteria. So bacteria they are also helpful in controlling oil pollution, and also they are helpful in mineral extraction. To extract minerals from their ores, by the action of certain bacteria, the mineral is separated easily, and that is one benefit of bacteria. So this is the benefits, and still there are several benefits. So just 2-3 benefits when we mention, that is enough. So this harmful bacteria, in case of beneficial nitrogen fixation, I said nitrogen fixation means rhizobium actually, when it is present in the root nodule, it will absorb the gaseous nitrogen, it will absorb the atmospheric nitrogen, and it will convert into nitrate and make available to the plants and help the plant to absorb, and thus it helps in the growth of the development of the plant. That is the benefit of the bacteria. Then harmful bacteria. Bacteria are also harmful. So that means the bacteria, usually the parasitic bacteria, they are harmful, and they cause the diseases. They are pathogenic, they are disease causing, and they are harmful bacteria. For example, the cholera, leprosy, tetanus, then plague, and many other diseases which are produced or which are seen in human beings, they are all by harmful bacteria. So harmful bacteria, they usually cause diseases like leprosy, tetanus, then tuberculosis. All these are some diseases. Even in plants also, bacteria causes disease. For example, citrus canker is one disease which is caused by bacteria. Then this is the benefits of bacteria, the harmful effects of bacteria, or in other words, diseases caused by bacteria. Mention the beneficial effects of bacteria. Short question, 2 marks. Mention the diseases caused by bacteria. So the diseases caused by bacteria means it is nothing but the harmful effects of bacteria. So that is about bacteria as an example of kingdom monera. And when we take the structure of a bacterial cell, definitely it is the structure of a prokaryote, which I have just mentioned. A prokaryotic cell which is having an indefinite nucleus, incipient nucleus, and no mitochondria, no Golgi complex, no endoplasmic reticulum, and ribosomes are of 70s type of ribosomes in case of prokaryotes. Then further, if you go into more detail, bacteria, they are also having small thread-like structures called as this flagella, and these flagella they are present usually at one end of the cell. So usually coccus bacteria don't have flagella. They are small projections which are seen on the cell, and they help in locomotion actually. So the bacillus bacteria also may be having, and depending upon the arrangement of the flagella or the arrangement number of flagella, again there is one more classification of bacteria which we are not concerned. That is about bacteria. Then coming to archaebacteria. So archaebacteria is another type of example. So this archaebacteria, they differ from the normal bacteria, and that archaebacteria, they differ in the cell wall structure, composition. So the difference between these are also unicellular, because already we have mentioned the monera character. So these are unicellular organisms, but they are not like this normal bacteria, archaebacteria. They are. So this archaebacteria differ in their cell wall structure from the normal bacteria, from this what we have studied so far. And another thing is, in this archaebacteria, they usually live in extreme conditions. So extreme means abnormal conditions. For example, in case of salty areas, they live. In case of hot springs, they are present. They are called as thermophiles. And in case of marshy areas, they are also present. They are called as methanogens. So these are the three types of archaebacteria. And so in very high salt containing soils, they are called as halophils. And in case of high temperature like hot springs, they are called as thermophils. In case of marshy areas, they are called as methanogens. So these are the three types of archaebacteria which we usually find. And then in the methanogens, so these methanogens, they also help in the production of biogas, what is usually used in the case of domestic purposes. So in the case of ruminant animals, these methanogens are found, and they are also present in the dung of the ruminant animals. So that biogas production is done by methanogens, archaebacteria. They are not normal bacteria, and these archaebacteria are different in their cell wall structure, and they are found only in extreme conditions, climatic conditions, highly salt conditions, and in high temperature thermophils. In case of marshy areas, we find they are usually called as methanogens. These methanogens, they are also taking or involved in the formation of biogas when the dung of the ruminant animals is used in the production of biogas. That is about archaebacteria. Then cyanobacteria. If you come to the cyanobacteria, now these are also unicellular, no doubt, but they are found in colony, or sometimes they are found in the form of filament. They are filamentous, in other words. So unicellular, found in colony, that means many cells, they form a colony, and they occur in groups, and sometimes the cells are arranged in a filament. So filament means suppose these cells are arranged like this, it is called as a filament. When the cells are found in groups like this, so when you see this structure, it appears as though it is multicellular, but it is not multicellular, and it is separate single organism, unicellular, but found in a colony, and usually such colonies are well protected by a mucilage layer or a jelly like substance, and in case of filaments, the cells are attached and they form a row or a single line. This is in case of cyanobacteria. And cyanobacteria are also having certain structures in their colony or filament which are called as heterocysts. So these heterocyst, if you take a single, it is a cell which helps in reproduction. So they are found along the filament here and there at different intervals, and the heterocyst cell helps in the reproduction of that cyanobacteria. And again, here there are different examples of archaebacteria and cyanobacteria. In cyanobacteria, if we take the benefits and harmful effects, it is also having some benefits and harmful effects. The example like nostoc, oscillatoria, and anabina, these are some examples of cyanobacteria. So anabina, oscillatoria helps in the nitrogen fixation like rhizobium bacteria. So when we studied about the beneficial aspects of bacteria, there we mentioned about rhizobium, which helps in the nitrogen fixation. That means the conversion of gaseous nitrogen into nitrate. Similarly here, certain cyanobacteria like anabina and oscillatoria and still many others, so there are some bacteria which help in the nitrogen fixation. That is one use of this cyanobacteria. And some harmful effects of this is, and another use of this cyanobacteria is, as the bacteria are found in colonies or filament, even when they are found in filament, around each filament you find a mucilage layer, protective layer. Even around each colony we find a mucilage layer. Even around the filament we find mucilage layer. So such filaments or colonies, when they are present in soil, so cyanobacteria is found in fresh water, marine, and even terrestrial or even in land. So and this archaebacteria also, I said, it is found in salty soils, hot springs water, and marshy regions. Cyanobacteria is also found in fresh water, in marine water, sea water, and even on wetlands. So when it is present in land especially, so this those bacteria, because of the mucilage which binds to the soil particles, so they help to prevent soil erosion. So the soil particles are not easily carried, they are not easily displaced from that particular place, hence they prevent soil erosion because of the mucilage. So they help in nitrogen fixation, they prevent soil erosion due to the presence of mucilage. And harmful effects, certain cyanobacteria, when they are present in water especially, and cyanobacteria are mostly they are floating on the water, planktons they are. So these cyanobacteria which are present, they, when they are present in a large population, when they rapidly increase and occupy most of the regions in the water, then they form what are called as algal blooms. So algal bloom means the water is completely covered by this algae. And one more important point is cyanobacteria, they are also called as blue-green algae. But other algae, they are different. We come under protista. The green algae, what we commonly see in ponds or lakes, so that is different. So this cyanobacteria is blue-green algae because it contains blue pigment as well as green pigment. So this because of the blue and green pigment, it is referred as blue-green algae. But the normal green algae what we see in the ponds or lakes like that, that is coming under protista. Then so when this algae is present in large population, it will completely cover the water, and to some extent it will release certain toxins and pollute the water. So by just observing the rapid growth of algae, one cannot take interest to use that water. So that means we don't go and have that water for any purposes. So that is why these algal blooms are produced by certain bacteria which are also called as blue-green algae. And this cyanobacteria, cyano means actually blue, a Greek word it is. So as they are blue and green in color, they contain a blue pigment called as the phycocyanin. They contain the green pigment, chlorophyll, and that is why they appear blue and green in color. That is why they are called as blue-green algae. Then another example of this, mycoplasma. So mycoplasma, it is a unicellular organism with containing many nuclei, several nuclei. And another important thing is there is no cell wall in mycoplasma. We find cell wall in cyanobacteria, archaebacteria and bacteria. So and this is one exception when we speak about the prokaryotes, and that is monera. Mycoplasma are unicellular and they are the smallest organisms. When we say which is the smallest living cell, we know that is a cell, but which is the largest cell, smallest cell. When we come across that, so mycoplasma is the smallest cell ever which is seen, and so these mycoplasma, they live in different conditions, in water or even in wet soils, and even when there is oxygen deficiency, they live in that type of environment. So where there is a scarcity of oxygen, suppose there is polluted water, so usually we don't find oxygen in that place. So in such places or in such areas, we find that the mycoplasma is present. So mycoplasma is one example which comes under monera. So this is about monera. So in monera we find bacteria, archaebacteria, cyanobacteria and mycoplasma. So that is about just a brief explanation of kingdom monera. Then if you come to the protista, just in brief, when we see protista, they are also unicellular organisms, but they are eukaryotes. So these are unicellular eukaryotes. So the main difference between this is in prokaryotes and eukaryotes. These both are unicellular. Monera is also unicellular, protista is also unicellular. But protists are eukaryotes, and they are also having autotrophic and heterotropic nutrition, as case of monera, where we are having both here. Here also the same thing, heterotrophic and autotrophic. And usually the locomotion we find here, there is a presence of pseudopodia, there is presence of this flagella, cilia are the locomotic organisms. And here we find the reproduction method is developed than that of the monera. We find sexual and asexual type of reproduction in case of protists. So in protists we find the developed organisms than that of the monera, and they are purely eukaryotes. That means the cells are having well defined nucleus, and they contain mitochondria, they contain endoplasmic reticulum, and they also contain Golgi complex, and the cells are well developed with the functions. That is about the protista. Then protista, they are again divided into 5 divisions. So these 5 divisions, they are chrysophytes, they are euglenoids, dinoflagellates, slime molds and protozoa. So about the different divisions of this protista, I will continue.